us?
Don't touch anything.
Say continue. Say,
oh yes, continue.
it's wonderful to see some of you in 3 dimensions. This is just incredible, terrific, lovely. And also wonderful to see other people via Zoom who are connecting today, because we have a special day today. Thank you for joining us for the Dissertation Defense Seminar of PhD candidate Jessica Taylor Martin. First of all, let me say welcome to the ancestral lands of the Muwekma Ohlone. I know that Jess is going to do a proper land recognition statement in a few minutes, but I could not begin without saying thank you to the Muwekma Ohlone, appreciation and admiration, and thankfulness for the fact that we work and live in their lands. I want to give a special welcome to Jessica's family, including Dr. June Taylor Fabrica Martin, Mom, Mr. Craig Thomas Martin. Fun fact, Jessica actually was born on the same month that her mother defended her thesis, her PhD thesis. Another fun fact, she also has a brother, Alex, and Alex and Jessica's sister-in-law is also Jessica Martin, not other Jessica. So too many Jessica Martins today. So it is a pleasure to introduce the speaker this afternoon. Jess comes from a very nice part of the planet, actually, from New Mexico. The place is an area that is beautiful because of the cultural diversity, culinary diversity, ethnic diversity, and of course, fantastic biological diversity. And I'm sure that must have been an inspiration for Jessica to study biology. And indeed, she went to the University of New Mexico, where she got her BSc in biology and also a minor in statistics. But more than taking classes and going to classes and getting a thesis and completing a thesis, she also interacted with fantastic people. In fact, 2 of my favorite ecologists on the planet, Professor James Brown was a person that she interacted with a lot. This is one of the most prominent ecologists in the country and actually on the planet. And also she interacted with Professor Felicia Smith, one of my favorite conservation biologists and mammalian ecologists. And so she comes with a terrific background from that fantastic group of people in the location. She actually did an honors thesis, and the thesis has to do with interactions between mesquite, that famous plant from desertic and semi-desertic areas of the planet, and interactions with rodents, which is very significant for me, because I am very interested in mammalian ecology and in rodents. And with that fantastic pedigree and her interest in mammalian ecology, and convergence that we're very much interested in music as well. We don't always coincide with preferences 100%, but we have a good degree of overlap there. But, you know, in some sort of a selfish manner, I was so delighted when she applied and she joined the lab because of all of these intellectual convergences, and including also artistic and personal common interests. As some of you know, her main fields of interest are disease ecology with a community ecology perspective, and the connections of that aspect with zoonosis, with an emphasis in rodents. And we will hear a lot about that today when she presents. During the time that she has been in the lab, we have had plenty of opportunities to interact in multiple ways. And so I am so glad to say this afternoon that I learned a lot from her and about her. And this has been very significant for me. Also, the time that she's been in the lab has given us plenty of opportunities to do some adventures together, some of them with happy endings, some others with not so happy endings. For example, in the area of happy endings, we have been in the rainforest of Southeast Mexico 2 times. And once for a class, Bio 375, she might remember that, probably. And another time when we went on an expedition with the entire lab, And because of that, we have two papers together we have published as a result of those interactions. One is in Journal of Ecology, the other one is in Biotropica. And this has been a fantastic set of adventures. And of course, we have had some more complicated attempts to do things together, such as, you will remember that during the past administration, the situation of many kids that tried to come across from the southern border to this country, they were detained in horrible camps, almost concentration camps. And Jess and I tried to bring to those kids some science and happiness and fun activities. We tried really, really hard to be able to go there, and we knocked on so many doors, but we were not able to get green lights to do that. So that's an adventure that I learned a lot from her, but it was not a happy ending adventure, but such is life. Still, I'm so grateful that I've learned a lot from her these years. Also, I want to say that Jessica shared a lot of literature with me, literature that is not only scientific, but goes to other aspects for me to understand the life of Homo sapiens, especially in these difficult times of life. I cannot go without saying that during the time of her PhD, she has encountered a number of very challenging situations, and so beyond the typical trials and tribulations of any PhD student. I'm so glad that thanks to her endurance and incredible spirit of perseverance, she has been able to overcome all of those challenges. And that has been really very important and even very emotional to me. And I'm so happy that she has been able to overcome all of those challenges, and we have her now in this situation coming today to do her dissertation defense. So I'm going to finish by saying thank you, Jess, for all that you have done for the lab and for me. And today she will share some aspects of her research. She will speak about bi-directionality in host-parasite interactions. And so without further ado, let me share the podium with Jessica. Thank you.
Thank you so much, Rodolfo. I wasn't expecting to get teary-eyed until the acknowledgement, so I'm good.
Jess, one quick note before you get started, a reminder that everyone on campus does need to be masked.
Yes.
Great.
Yes, everyone in the audience is masked. We're good to go.
Yourself included. As our first hybrid defense, I know this is not ideal. I know the science says otherwise, but that's what university policy says. So thank you so much.
Is it okay if we stop recording and then I don't wear a mask? I feel very strongly about this.
Regardless of recording status, university policy does say that everyone needs to be masked. I understand that science says otherwise right now, but university policy is saying that. So I apologize. Not ideal, but we are still all really excited to hear you defend. So thank you. Thank you.
Okay, well, CDC be damned, we'll do this defense. CDC guidelines be damned. All right, well, with that, Rodolfo already did a beautiful job as a land acknowledgement to the Manlaqualoni, and I just wanted to emphasize that they are still a group in the Bay Area, and you can check them out on their website to learn more about that indigenous group. I also wanted to acknowledge the place where I do my fieldwork in Arizona. This is a beautiful picture taken by my father, Craig Martin, and this is the land that the Chiricahua Apache lived in before being occupied by Spanish and later Anglo folks. And with that, I would like to hop into the science I'll be talking about today. As Rodolfo mentioned, we'll be talking about bidirectionality and host-parasite interactions. And returning to, I'll begin the story with talking about my fieldwork. Oh, I should move, okay, zoom on in Portal, Arizona. So these illustrations are done by my dear friend and colleague, Callie Chappelle. And right now we are seeing some of the community of mammals that were my focus for much of my PhD. And we will first be looking at the impact of the mammal host community on parasites, in this case, fleas and ticks. So this is what my artwork looks like compared to Callie's. So we're all aware, especially in the last year, that pathogens and parasites have huge effects on humans. So we have the rabies virus that can infect all species of animals through bites. We have malaria, which is a huge deal in the tropics and other areas that Aaron Mordecai knows very well, that's transmitted by the bites of mosquitoes. We have the common cold, which looks a lot like a virus that's been affecting us all very heavily the last year and a half. Cholera, which is a waterborne disease, and then Giardia, which if you go camping or backpacking, you'll know that you need to filter your water, or this is a risk that you'll have. So to start off with a few definitions. First, vector-borne diseases are those that are transmitted by blood-feeding arthropods, such as mosquitoes and ticks. Second, zoonotic diseases that are caused by pathogens that infect humans as well as other species of animals. Getting water is going to be awkward, but I'm doing it. Okay. Oops. All right. So for this defense, I will be focusing on, especially in the latter half, on tick-borne zoonotic diseases. And ticks actually cause the majority of zoonotic diseases in zoonotic tick-borne disease cases in the US. And many of these tick-borne diseases, aside from Lyme disease, are poorly studied and underdiagnosed. And these are especially in areas of the world like Africa and South Africa. And diseases are really difficult to manage and control. As an example, I'd like to talk about Lyme disease, which is the most infamous tick-borne disease. The vectors are ticks of the genus Ixodes, and the pathogen is a bacteria named Borrelia burgdorferi. So we have the tick vectors, and then the hosts for the immature ticks, larvae and nymphs, are deer mice. And then those ticks, once they take a blood meal, drop off, and the adult forms feed on deer, which are very prevalent in the eastern US, which is part of why this is such a problem. And then when people are bit, that causes Lyme disease. And this is a telltale sign that you've been infected with Lyme disease. It's a bullseye target, and you need to get yourself to the doctor if that's the case. So we've talked a bit about how pathogens impact their hosts. And what I really want to emphasize here is that this relationship is bidirectional, in that host communities can also impact their parasites. First, a very famous debate that I will not get into unless asked about is the relationship between species diversity, simply the number of species in a community, and disease risk. There's a paradigm that when you have more species in a community, that can actually protect against disease risk for humans. Part of this is because of community composition, and that's just the identity of species in a community. And the reason that community composition can be important is because there's variation in what's called host competency. And this means that some animals, species, and some individuals even are better at spreading disease than others. So if you're a very competent host, then you're one that's going to be spreading a lot of disease. One thing that I am fascinated by, but will not be touching upon today, is immune functioning among individuals as well as species. There's a paper that I think is really fascinating called Living Fast and Dying of Infection, that talks about how rodents that have a life history strategy of reproducing lots and lots of babies actually have a trade-off where their immune systems don't function very well. Okay, water. Sorry. All right. So with the first part of my talk, I'm going to be focusing on how host communities impact their parasites. And I'll be looking at that through the study at Portal, Arizona, in terms of mammalian host segregation altering ectoparasite infestation in a desert ecosystem. The second part of my thesis I'll be talking about is thinking about the consequences of tick species traits on their hosts. So this first chapter that I'll be talking about was in collaboration with Richard Grewell, who's in the audience today, Roberto Losapio, Rodolfo, and other collaborators that I have been working with at the University of Florida. The first question that we're addressing in this study is, how does the experimental removal of rodent community dominant kangaroo rats affect infestation by fleas and ticks? I'll be talking about the methodology of this in a moment to explain that a bit more. And second, how does spatial segregation between kangaroo rat and jackrabbit hosts affect tick infestation? Portal is a long-term research site. I was so pleased that Rodolfo mentioned one of my favorite ecologists, one that I much admire, James Brown, who established the site with other folks in 1977. And every month they've been going there and capturing rodents and sampling them, and it's capture release. So they've been catching rodents there and for a long time. And this is the location of it in the Chihuahuan Desert near the border between Arizona, New Mexico, and Mexico. So for the first question about what happens when you remove kangaroo rats from the community, we're going to be thinking about a dichotomy here. So there's specialist parasites, which are jackrabbit ticks, and then there's generalist ectoparasites, which are fleas of several species we found, dominated by a generalist flea species. In terms of our expectations, when you remove a host and the parasite is a specialist, then you don't expect to see the parasite. Likewise, when you remove one of the hosts of fleas, you are not going to see fleas on that particular host. However, what we were really interested in looking at in this study was what happens to the rest of the rodent community, and how does that translate indirectly into changes in the flea community? For the second question, thinking about spatial segregation, habitat use between kangaroo rats and jackrabbit hosts and how that affects the specialist tick infestation, we have these three different treatments. So we have unfenced plots where the kangaroo rats and the jackrabbit hosts can overlap in their habitat use, rodent access plots, which are fenced, I'll be showing that in a moment, and then finally kangaroo rat removal plots where there were no kangaroo rats present. This is the experimental design of our study site. You can see this is a Google Earth view. There's faint outlines of all these fenced areas, which are 1/4 hectare in size. And we have the unfenced plots in green. Oh, I could use this, but anyway, not gonna mess with that. Unfenced plots in green. In yellow, we have the rodent access plots. And then red are the areas where kangaroo rats were excluded. I already mentioned that we've been doing monthly field sampling here. And just to kind of clarify what the setup is with the fences are about yay high off the ground. And this is a picture of a rodent access plot. So the gates are about this big, and all the rodents, including kangaroo rats, can move in and out as they wish. In the kangaroo rat exclosure plots, they're about this big, and the kangaroo rats have such big heads that they can't fit through the gates. Thus, they are excluded. So for 40 years, people have been looking at small mammals at this site. They love their mammals. And in 2015, I reached out to establish a collaboration with these folks at University of Florida because they had never considered ectoparasites. And as Rodolfo knows very well, rodents are really gross creatures. They have a lot of parasites. They spread a lot of disease. So we wanted to take advantage of that and hop on what they were already doing and sample for ectoparasites. So we've been sampling between 2015 and 2019. A picture of a flea comb. It's what you'd use on your dog or your cat to comb fleas into a basin. And then we also subsampled ticks as well as counted the total number of ticks on each individual. So all in all, we wrangled almost 700 rats. Richard and I did a great job with that, of 12 species. And we collected about 350 fleas of 10 different species, dominated by a generalist species, and then collected 378 ticks out of about 1,000 that we observed on tick hosts. All of these turned out to be Dermacentra peruma or jackrabbit ticks. In terms of statistics, I'm not going to get into the details for you all. I wanted to let you know what we were measuring. So the response variables in this model that we used, generalized linear models, we looked at prevalence, which is the proportion of rodents that were infested with at least one flea or at least one tick. We looked at them separately. Second, we looked at intensity of infestation, which is the number of parasites per individual, basically per capita fleas or ticks. In terms of predictor variables, we were primarily interested in the treatment, whether the plots were fenced or unfenced, had kangaroo rats or not. We also looked at a couple different years. We were interested in the rodent host species because of potential variation in host competency. And finally, we looked at body mass, which is not something I'll talk about today, but happy to answer any questions about it if they come up. Gotta not put my water there. So, no, that blocks the camera. Okay. To answer the first question in terms of generalist flea species, we found that the infestation by these flea species was actually independent of kangaroo rat removal. So although one host was lost, we found that there were some species in the rodent community, obviously, that came in and filled the niche of the kangaroo rat. They were more popular or more abundant relative to other species. But overall, everything came out in the wash, and we didn't see changes in the prevalence or intensity of infestation of the rodent community as a whole. We did find that there was variation in rodent host competency, and I get to talk about one of my favorite species, also the grossest. I would recommend everyone check out this YouTube video called "The Grasshopper Mouse is a Killer Howling Rodent." Yeah, they really do howl like wolves. So they had the highest degree of flea infestation. So there were some traits that they have as species that made us think this is as an explanation of why they had the most fleas. First of all, the kangaroo rats themselves are specialists and eat mostly seeds. The pocket mice, the version of kangaroo rats, also eat mostly seeds. And these guys actually prey on other mice, in some cases, even eating kangaroo rats. And they also share burrows with other species. And both of these are opportunities that parasites would be spread. So if you're eating a dead kangaroo rat, then those fleas are gonna hop off onto you. If you're messing around in the burrows of other rodent species, that's another time that you may get fleas. As I mentioned, the ticks in this system in particular, are specialists. So the immature ticks are found, the larvae and nymphs were found only on kangaroo rats. And these horror movie ticks, they're not actually that big relative to jackrabbits, are found on jackrabbits, which we did not sample directly, but in a little bit, you'll see what we did to sample their activity levels indirectly. So the results for ticks were in strong contrast to what happened with the fleas. We found that in unfenced and transect sampled outside of the plots or in unfenced areas, we saw 83% of kangaroo rats were infested with ticks. In contrast to this, we had 38% of kangaroo rats with ticks in the rodent access plots. And then when you don't have kangaroo rats, we didn't see any ticks. So we also found tick intensity of infestation, or the number of ticks per individual, was higher outside of the study plots. So I wanted to point out a couple of things here. One, we maxed out at 30 ticks on one kangaroo rat, and that was in the area outside the plots. And we saw this pattern across both years. I'm going to focus on November 2016 because we had a higher overall infestation that year. And in contrast, in the rodent access plots, we found a maximum of 10 individuals. And you can see the means are also very different, significantly different, according to our models. In the unfenced plot, 16, we counted a total number of 338 ticks on 53 kangaroo rats. In contrast, we had a smaller sample size outside the plots. So we caught 20 kangaroo rats and 29 ticks. So overall, we had 11 times more ticks observed outside the plots. And this had us puzzled for a while, to be honest. And this was the point where we decided to start looking at jackrabbit activity to see if the presence of the host of the adult ticks could explain these results. So we did what any good mammologist does, or any mammologist really, and looked at poop. So we did these fecal pellet surveys, and the quad rats are about yay big, and we collected all the poop in the quad rats and then measured them and identified it to be jackrabbits, and then found that we had higher activity levels of the jackrabbits outside of the study plots relative. So they're fenced, as I mentioned. And before we started looking at this, we thought that they didn't ever get in the plots. Turns out they are active there. Every once in a while they jump over the fence, but overall they're less active. And we also did camera trap surveys. Hold not a lot of pictures, and found that although they are present in all the treatments, they spend most of their time outside the plots, which makes sense in the unfenced areas. So we did find ultimately that increased spatial segregation between these tick hosts decreased tick infestation. And the mechanism for this is well documented in the literature. And the mechanism is that encounter rates were likely decreased between ticks when the hosts were spatially segregated. So as I mentioned, ticks take blood meals at each life stage. So the immature ticks are on the kangaroo rats. They take a blood meal, drop off their host, grow into adults, take a meal, drop off, lay eggs. And that cycle, when the habitat uses overlap between those host species, that results in higher tick infestation because they're more likely to encounter hosts. So social distancing works for rodents as well. A friend of mine said that when I practiced this talk with her. So in sum, we found that specialist ticks were absent when we removed kangaroo rats. And when you remove kangaroo rats, they're not one of the hosts. And overall, generalist flea infestation was independent of removing the kangaroo rats. So we found ultimately that kangaroo rat removal differentially impacted generalists and specialists. To return to the concept of how host communities overall affect their parasites, we did look at community composition in this study. And we found that when you remove one community dominant rodent, parasite infestation was altered, and this is different between generalists and specialists. We also found variation in host competency. So we found those grasshopper mice were the most competent flea hosts. And we also found that differences in host habitat use or spatial segregation affected flea and tick infestation or tick infestation. In terms of implications for the health of human and wildlife, we found that as a general, there's a general pattern that specialists and generalists respond differently to changes in their host community. We know that host and parasite communities are all impacted by anthropogenic factors, such as climate change, land use change, deforestation. The list could go on and on. And we also know in the literature that there are winners and losers. And not only are there winners and losers in terms of hosts, we can also think of parasites and pathogens. And ultimately, this has implications for how to prevent diseases as well as manage them. So from here, I'll be shifting to another chapter of my dissertation, where I collaborated with Ilya Fischhoff and Barbara Hahn at the Carey Institute of Ecosystem Studies, located in New York at the epicenter of Lyme disease. I'm looking at, in this study, we're looking at the consequences of tick traits on their hosts. So connecting the traits of individual tick species to their vector status or ability to spread disease to humans has the potential to identify new vectors. And that's something that we are looking at in this study. So I mentioned Ixodes ticks at the beginning, and though that genus of ticks are the poster child for Lyme disease. And it turns out that dermacentor ticks, the same genus as the jackrabbit tick, although not looking at the entire genus, spread different diseases than Ixodes ticks, and they are less studied. So the primary pathogen known that dermacentor ticks spread is Rickettsia bacteria, and those can cause spotted fevers, most famously in the US, Rocky Mountain spotted fever, also African tick bite fever, and typhus. And these diseases are much less studied and underdiagnosed likely than Lyme disease cases in the United States, where we have a lot more resources. The questions in this study that we looked at were which traits of these dermacentor tick species best predict their status as zoonotic disease vectors, so diseases that can be spread between humans and other species of animals. And which described and understudied dermacentor tick species are most likely to be vectors of zoonotic disease. In terms of the data we collected, we looked at 76 different morphological characteristics, such as body size, the length of their body mouth parts. We also looked at host range on the species scale. We looked at the number of host families and the number of host orders. We also took a global view and looked at the geographic distribution of each of these 42 dermacentrous species. And we looked at ecoregions and climate zones, which I'll explain in a moment. To determine the status of these ticks as whether they're known zoonotic disease vectors or not, we used a database called Gideon, the Global Infectious Diseases and Epidemiology Online Network, as well as reviewed over 500 journal articles and books to collect data on their distribution and morphological characteristics. We found that about a third, or 13 of the 42 known dermacentre tick species, are known to be zoonotic disease vectors. In terms of the geographic distribution, we had about 4,000 location data points for all of these species. And here is showing 2 examples, the distribution of the moose tick, Dermacentre albopictus, which is a known zoonotic vector species, as well as Dermacentre reticulatus, which is the ornate cow tick, which is also known as a vector species. And I would like to give a shout out to iNaturalist, which is a crowdsourced database by citizen scientists where anyone can go out and observe plants or ticks in this case, and all that data, a lot of it came into our geographic distribution data. So to show you a little bit about what we did is we took those 4,000 data and condensed into counting the number of biomes and ecoregions and Köppen-Geiger climatic zones. So this is showing the globe and divided up into 8 biogeographic realms and 14 biomes. Some examples of biomes are tundra or boreal forests or subtropical forests. There are 867 terrestrial ecoregions on the Earth. And so we were counting the number of ecoregions that each tick species was found in, partially to think about how variable the environments are that they can survive in. I will also not get into the mechanics of this until I talk to my committee in more detail. Machine learning is a big bucket, but the particular technique we used is called generalized boosted regression. And the outcome of it is that we identified and ranked the relative importance of each of the predictor variables we measured. Then, using we were able to predict new vector species based on their traits profiles of the known vectors. Here's a blank graph so that I can introduce the axes. The relative importance shows how much the amount of influence of each variable in the model. And then on the y-axis, we have each predictor variable. So what I have is color-coded by category. So first, the top is citation count in red, and that's about in the middle, which is actually a good thing. So citation count was just the number of studies that were published on a particular species. So if this had been by far the most important, that would be telling us that it was just study bias and there wasn't a real biological signal. So instead it came out to be less than 10%, about 5% relative importance. We had the top 2 predictor variables had to do with host range, number of host families, and number of host orders. We then saw that the geographic range, the number of climate zones, and number of biomes were important. We found that whether or not they infested humans was important, and whether or not they spread disease in humans. And then we started to find morphological characteristics. We looked at 76 of them, and you can see that body length and width of male ticks were the top 2 predictors. We then had number of ecoregions, citations, and then a lot more morphological characteristics that were of lower importance. I'm going to show a couple of plots that are called partial dependence plots that are a bit complicated, but conceptually, I think I can help you make sense of them. So we found, as I mentioned, that dermacentre species with broader host ranges were more likely to be zoonotic disease vectors. So if you look at this plot, you see number of host families on the x-axis and then proportion on the y-axis. So the proportion, the gray bars are a histogram showing the actual distribution of how many host families tick species have. So if you see the first bar shows that the majority of tick species have very few host families. And the black line is showing zoonotic vector status. And you see this inflection point around 9 host families. So as soon as a tick species has about 9, then it's much more likely to be a zoonotic vector. We also found that dermacenter species with larger geographic ranges were more likely to be zoonotic disease vectors. So I didn't show a picture earlier of the climate zones, a little less overwhelming than the ecoregions. There's about 30 globally. And we saw a similar pattern where most tick species had lived in a few number of climate zones, and then you see a steep inflection point, and if they live in a greater number of climate zones, more likely to be disease vectors. Finally, we found that dermacenter species with larger bodied male ticks were more likely to be disease vectors. This is a result that we found actually puzzling and are still thinking about in terms of tick biology. The female ticks will take one large blood meal and they'll use that to produce their eggs. The male ticks, the adult male ticks, take less frequent and smaller blood meals. So we're still thinking about how body size plays into this and are excited to keep exploring these results. So in terms of a traits profile, we wanted to think about how the interplay between these 3 categories worked. So first, morphological characteristics can affect the ability of a tick to find new hosts. There are sense organs on the ticks that they use when they're questing, and if those sense organs are larger, they are more likely to be generalists because they're less picky in detecting the smell of their hosts. Morphology can also impact a tick's ability to survive in variable conditions. Perhaps if they're a larger body size, they're able to inhabit a broader geographic range, easier to overwinter. And finally, larger geographic ranges can feed back into host range. So if a tick is distributed widely, it's more likely to encounter more hosts. And when you have more hosts, those hosts move around, and there's a positive feedback loop between geographic range and host range. And ultimately, all those factors come together to determine to influence whether a tick is a vector of disease in humans. In terms of predicting new vector species, there were a lot of species that were described in the last 10 or 15 years, particularly in Southeast Asia. We found that dermacenter pavlovskii had a greater than 50% probability of being a zoonotic vector species based on its traits profile. We found that its known geographic distribution is in China, and this database is just collected in one paper. And so this is the known distribution of this species. Obviously, there is sampling bias, and that's something I can answer questions about later if folks are interested in more details. But it's distributed widely enough that it had the traits profile that reflected that it was likely to be a vector. We also found that it had a relatively broad host range, despite not having a lot of publications. Relatively few citations. We had 33 journal articles published on that versus dermacenter marginatus, which has more than 1,000 and is a very well-known disease vector. In terms of future directions, we're really excited at the possibility of taking this geographic database and doing species distribution modeling. A lot of species distribution modeling is happening right now, and this is an example of one of those being species distribution modeling for the dog tick, Dermacenter variabilis, and it's actually overlaid, the habitat suitability is overlaid with cases of Rocky Mountain spotted fever, one of those rickettsial diseases I mentioned in the beginning. So with the database that we have, we're actually able to map multiple tick distributions and potentially overlay that with host distributions and look at disease incidence. So we have all kinds of possibilities with this database that we've built. So to return back to the broad theme of tick-borne zoonotic diseases, many tick-borne diseases transmitted by dermacenter ticks are poorly studied and underdiagnosed, especially in areas such as Southeast Asia, where there's a high diversity of dermacenter ticks that are not well studied. As I mentioned, tick-borne diseases are very difficult to manage, and the Lyme disease system is a particular example of very well studied, and they're starting to think about how the host community can be altered to ultimately impact the disease risk. Finally, we have limited time and resources to devote to tick-borne diseases, especially in the last year. And in terms of public health significance, something that we would ultimately like to do with the data is think about determining disease risk hotspots that can be used for monitoring and prevention. This is an example of a map from my collaborator, Barbara Hahn at the Carey Institute, looking at global hotspots of zoonotic mammal hosts, thinking about their diversity, and being able to map hotspots of disease risk across the globe. Ultimately, one of our goals is to generate scientific knowledge that we can share with public health officials. So to recapitulate, host communities can impact their parasites. Understanding the ecology of communities contribute to our ability to control and manage zoonotic diseases. We saw how community composition at our field site influenced the prevalence of generalist and specialist ectoparasites. We saw there's variation in host competency. And finally, we found that habitat overlap was able to affect the parasite infestation. We also found that in the other direction, parasite species traits do affect their zoonotic disease potential, and determining trait profiles of existing vector species aids in predicting future disease risk. And there's a lot of work being done by my collaborator, Barbara Hahn, as well as others, in thinking about zoonotic vector species and rodent hosts and a wide variety of species that are contributing to disease risk. And with that, I would like to thank you very much and ask for any questions. Yes, I see some chats. Oh, I see a lot of chats.
Well, great presentation, Jess.
Thank you, Richard.
One general question.
Mhm.
I think the two pieces you presented, and that is with habitat change
Mhm.
And maybe separation of species. Do you expect the predominance of either the generalist or specialist type of dermacenter tick
Mhm.
species? And then what does that mean for future zoonoses?
Okay, so let's see if I can repeat Richard's question for the Zoom folks. Richard is interested in the concept of generalist and specialist dermacenter species, is that correct?
Mhm.
And the implications of being a generalist versus a specialist. And I would say that ultimately, especially as we see with like rodent reservoir species, ones that can spread zoonotic disease, that the ones with the generalists tend to be winners, as in they spread more disease. So I would expect that dermacenter species that have those traits that allow them to have a wide geographic distribution, allow them to have a wide degree of hosts, all of that contributes to their ability to jump into one more host that we especially care about, which are humans. Thank you. Yes, there's 30 chats. So now we got to figure this out again. Let's see. Oh my gosh.
Jess, I can read them to you if that's helpful.
That would, yeah. Filter out the congratulations.
Perfect. Okay. So we have one from Elsa. Do you think enough people are working on this? And is there enough funding to follow up on further building your database?
Yeah. COVID is taking up a lot of resources, as we all know. And unfortunately, tick-borne diseases are not going away. And there's many factors that could lead to them increasing in incidence. Aaron Mordecai on my committee has looked at mosquito-borne diseases and how climate change can differentially impact, where some areas may have more disease, some areas may have less under future climate change scenarios. But yeah, I don't think it looks great because funding is limited right now. And as I've hoped that I've emphasized to the audience, tick-borne diseases are important and not going away and could definitely use more resources dedicated to them. And I'm excited about being one of those scientists that will continue to work in that area.
Awesome. Elsa also asked, how did you choose this topic?
How did I choose this topic? Wow, that's a deep question, Elsa. I guess, I think it's a combination. I guess it kind of fundamentally returns to what my values are as a person. I followed what was intellectually stimulating for me, something that I thought was, that I can get excited about intellectually, even though from an outsider's perspective, it's really gross. On the other side, I really want to be able to make a positive impact in the world in my life. And it turned out, my path is circuitous, but ultimately I feel like working on public health issues is something that I'm really passionate about. And I'd like to continue to work on that in my future jobs and career.
Excellent. We also have one from Eric Douglas. Yay, great talk. I was curious about the difference between geographic range versus host range.
Yes. My apologies if I didn't make that very clear. So the geographic range is literally, we took data points of a tick was reported here in Wyoming. A tick was reported here in China. And then we collected all those data points and put them together to say, okay, this tick species is located in coastal California or this tropical area in Mexico. And then we added those up to have an approximation of how widely distributed a species is. And then host range is simply the number of, as we looked at it, the number of host families or host orders, in this case, mammals. I hope that answers your question, Eric. Thank you.
Let's see if there's questions.
Yes.
Anybody have any questions?
Yes, Erin.
I know I'm gonna get to ask you more later, but I was curious whether you included any traits that related to human and tick contact rates. So like something around the propensity to occupy edge habitats or parks, or whether it was just sort of like, is a tick known to be found on humans or not?
Sure. Yeah, that is stuff that I've learned from talking to the Santa Clara County vector control folks. So those are things that they're looking at on this very micro scale. And I think that's really interesting, and there's a lot of potential there. And with this study, although we had a lot of depth in terms of looking at specific morphological characteristics and depth with the geographic distribution, we didn't really think about those like microclimate things or how likely a tick was to be desiccated if the humidity is low. But it's all exciting stuff. And I think it could be grown out to combine with that kind of data. Yeah, thank you.
Your turn.
Oh, my turn now. Anything else, Lorena?
We
Sorry.
just one, can you repeat the question? I didn't really hear Aaron's question from.
Yeah, that was from Julio, who's on my committee. Aaron was asking about whether we looked at or considered variables that could think about the encounter rate between humans and ticks in terms of like whether they occupy edge habitats or are, I don't remember what else, mostly edge habitats. Yeah.
I got it. Fantastic. And I heard the answer. Thank you very much.
Okay. Thank you, Julio.
You have more questions?
Lorena, are there any more questions?
I think we see one last one also from Elsa. How do you get to continue studying this at Stanford or where else potentially?
So I haven't announced it publicly, but I have accepted a postdoctoral position in New Mexico. Yeah, thanks. Getting Zoom thumbs up from Callie in the back. We can't undo that. Yeah, so I'll be heading in August to New Mexico State University and continuing to study diseases and mammals. Thank you for all the congrats in the chat. Okay, so.
Perfect. I believe that was the last of the questions on Zoom.
Okay, we have one more from the live audience. John Alberto.
So if you consider the relationship between jackrabbits and the kangaroo rats in a lot of way, how would you write down the model and how considering the ticks actually would change that?
So John Alberto is asking a technical question about Laca-Volterra models, which have to do with predator-prey interactions. But in this case, you're thinking in the context of the ticks as the predators and the mammalian hosts as the prey.
You know, it can be also just competition.
And then within that framework, what was your question?
How ticks, how disease will change these interactions and our interpretation of data.
that I understand your question in that context. I mean, there could be cases where there could be an epidemic that wipes out a host. And then when you would see, for example, part of my postdoc funding is to look at rabbit hemorrhagic fever virus, which is not spread by ticks. But if you have another disease come and knock out the hosts, then you would expect to see less ticks and have that reverberate through other hosts. Does that answer your question partially? No, not at all. Okay.
And how the tick might modify the outcome of competition.
Yeah, competition. Yeah, I mean, if we're thinking about like host competency and competition, host competency. Yeah, so if there's a species that's more vulnerable to ticks, then you would expect that to have a greater impact. Okay.
Yeah.
Lucky New Mexico. Okay, any other questions? Rodolfo?
Any questions there?
Many congratulations. I think Lorena, are we good with questions?
Looks good with questions and from Zoom.
Okay, I'm gonna stop my share.
Okay, any other question from here? Okay, yes, I think we have some other slides to show.
Oh my gosh, I forgot about acknowledgements.
Mm.
Shoot. Okay, okay, one moment. I forgot, this is the best part. So in my fashion as a musician, I have, oh, not at the beginning, but there will be some music in the acknowledgements, and I need to exit the slideshow and go to my other secret slideshow. Okay, we have, oh, wait, Zoom. Here, share screen. Clearly, I have not been TAing classes in the last year because I'm like not that great at Zoom. Okay, start show. We see everybody. Okay. First, of course, I would like to thank Rodolfo, my advisor, and he will have a few moments of acknowledgements in a few moments. And I would also like to thank Erin Mordecai, Julio DeLeo, Tad Fukami as members of my committee, and David Schneider, who has kindly agreed to be the chair of this committee. There's an immunology. Second, I'd like to acknowledge all of the wonderful biology department staff, Claudia Ortega. This is going back years now because I've been here a long time. Claudia Ortega, Dan King, Vanessa Sutter, Samantha Schwartz, and Lorena Rodriguez. Thank you so much for your help over the years. I'd also like to acknowledge Mary Beth Mudgett, Jose Janeni, and Martha Sayert, who have helped continue my funding so that I could make it here today and do this. I have several funding sources and I'm grateful for all of those. This is a lot of pictures, and this is only the current Dearza Lab. So I've really valued being part of the Dearza Lab and this community over the last 7-ish years. We've had lots of folks come and go. I've not forgotten you, especially if you're on Zoom today. And I just, I think the pandemic has been an interesting time for Dearza Lab, and I really have appreciated the time that we've been able to take to just check in with each other on a more personal level. And that wasn't something that happened so much before. And I found that really special to have the young people, as Rodolfo calls them, also be part of the lab, the co-terms and the undergrads and the recent grads. And yeah, this is a very recent picture when we got to help Lucas and Tyler do their bird catching. I would also like to thank my collaborators at the University of Florida. Morgan Ernest is the PI on the Portal project, has been running it for many, many years. Grad students, now former grad students, Ellen and Erica, as well as Glenda, who runs the Portal field site. At Stanford, we've had Richard Ernest Ruel IV. I'm sure he loves when I say his name. It's just too good, Richard. John Roberto, who helped with the stats, as well as Rodolfo. And my lovely parents, who will have a slide in a few moments, who actually collected rabbit poop and looked at pictures and did a lot of wonderful things, and I couldn't have done it without them. My lovely collaborators at the Carey Institute, Barbara Hahn, Ilya Fishoff, and Adrian Castellanos, who I have not met, but who wrote a lot of custom code for the machine learning bits. And I would also like to give a shout out to giants in the field, power couple Rick Ostfeld and Felicia Keesing, who have inspired me with their science. And I got to meet them in person. And Rick was the one that suggested we look at rabbit poop. And it honestly rescued me from a pit of despair of that project. So they helped me turn things around. And I hope they hear my thanks at one point or another. Okay, oh boy, now I have to really do this. All right. Oh man. Okay, so Rodolfo, man, this feels like some weird like boombox outside the window thing. This is Rodolfo's favorite Beatles song, and I wanted to say how inspired I've been by you as both a person and a scientist. You look like a complete badass in this picture. I couldn't help but put it here, holding this howler monkey, probably in the 80s. Rodolfo mentioned that we went to Veracruz together on multiple occasions, and I got to learn from him as a field biologist, knowing all of the plants. And I also wanted to include, a picture of him with an elephant working in Kenya, which is a study system that very much inspired me. And then he also talked about outreach and trying to help spread science to those that have less access to it. And that's really inspired me. And it's something that I am going to continue pushing for going forward in my career. And a lot of that is thanks to you. Okay. I don't know how to transition gracefully. So this is this is for Richard. Okay. Yeah. So Richard, you have just, no. You've been a rock star collaborator, and I just really wanted to thank you personally with this song called Dang Me by Roger Miller, which somehow became our like field theme song. So dang, it's been a journey and it's not over because we got to publish those papers this summer. Yeah, we were out in the field together when Trump got elected. And now that was a day, and we had a day where we had this many rats and traps, and we had a day, and we made it through. And thank you so much for all that you've done for me, Richard. Whew. His hat? Oh yeah, the hat. Yeah, I also made Richard handle all the wood rats because they're scary, even though I did that in my undergrad thesis. All right, so then, Roger Miller isn't over. Okay. This is so funny. Okay, so I wanted to thank my friends and family now. So cheesy. I recorded this song for Jeff for his birthday, but he's in a few slides. Yeah, just all the folks, like, as I've said, I've been here. There's been a lot of data turnover. And these are some very old friends, some family, some of whom are no longer with us. Wanted to acknowledge those friends. And also newer friends, my long lost cohort who graduated mostly in 2017. Alana is here in the audience representing. Thank you so much. I've been able to play music with my band, with Jeff and my friend Mitch. I wanted to acknowledge my church community, the Unitarian Universalist Church of Palo Alto. They allowed me to play my agnostic gospel music in their sanctuary as, yeah, and then that's my D&D character. And so I've been living in a co-op for the last 3 years named Bubba Land, currently occupied by myself, Callie, Jazzy Jeff, Megan Shea, Hope, Noam Hart, and it's just been really wonderful and a very safe place for me to be for the pandemic. So thank you so much. Almost made it through without crying. Okay. Woohoo! A shout out to Callie and Jeff. It's been difficult. Yeah. Rodolfo really doesn't want me to cry for a long time. Okay, it's been difficult and you've been there and you've been rocks and I just really appreciate it. Okay. Almost done, I swear. Whew. This song's almost over, so I can let it play out. I also wanted to thank my parents, of course. Couldn't have done it without you either. I'll be more eloquent maybe later when I'm not. Okay, anyway, this song means a lot to me, and love you guys. Whew, okay, now I'll be done. I'm gonna play myself out, folks. I've never seen anyone do this, but thanks, Jess, for doing this. Thank you. Thank you so much for being here on Zoom or in person. And I'm playing myself out with a recording of the Bare Necessities that I made a couple years ago. Okay. Bye. Thank you all so much. Yeah, you have to see her awkwardly. Yeah, well, I guess I have to walk out, yeah. Oh, I'm back. Okay, I'm back on the regular Zoom camera. Tears in our eyes too. Oh, thank you so much.
Way to go, Jess.
Thank you. I'm so fucking proud of you. Oh my God, I'm sorry. Oh my God. Good luck. Good luck. It'll be fun. You're gonna go take John off after that question. Thank you, Lucas. Yeah.